Welcome to Melanoma Insights for Professionals, brought to you by Melanoma Institute Australia. I'm Professor Georgina Long, medical oncologist and medical director at Melanoma Institute Australia. Welcome back to our special Vodcast series, Redefining Immunotherapy, Lessons from Melanoma. This is Episode 5, where we look ahead to the future of immunotherapy, separating hype from reality and exploring some of the most exciting emerging frontiers, including cellular therapies, mRNA-based approaches, and personalised cancer vaccines. If you haven't listened already, I'd encourage you to go back and listen to our earlier episodes where we discuss how immunotherapy works, how we can make better treatment decisions, how to recognise and manage toxicity earlier, and how evolving strategies in melanoma may inform practice more broadly. Joining me in the studio today are my medical oncology colleagues from Melanoma Institute Australia, Professor Alex Menzies, Associate Professor Mateo Carlino, and from Portugal we have Associate Professor Inês de Silva. We're back at it again, talking about our favourite topic, which is managing cancer, particularly melanoma. So hype versus reality. What is the future of immunotherapy in melanoma? Because we know melanoma is the leader in this field and therefore impacting other cancers. So, I'll throw it to you, Alex. Have we got there yet?
Not at all. We're scratching the surface, to be honest. I think, it's amazing that, for example, blocking PD-1 can cure half of patients with melanoma. We don't really understand why it does that. We don't know who respond. We've got a lot more to learn about the drugs we have now, how they work, why they don't, how to use them in better settings, who needs single, who needs combo, etc. But it's really exciting because there's clearly a whole lot of other treatments coming down the pipe, again, often pioneered in melanoma, because whilst it's wonderful and we say, look, immunotherapy can cure half of our patients, for half of our patients, they might have finished all standard therapies after one dose of one drug and you're finished and you're done. You could be 6 weeks in and be told you're done.
Yeah, you're
Whereas
cured.
in other cancers, there's other drugs. They may not be curative. They may have limited benefit, but there's other options.
But do you think we're curing everyone we can possibly cure with our current tools? I think we're trying hard. We've had 15 years or so of trying to nuance it. But I wonder if across cancer they're
I think
doing
we've got
this.
a hint about how poorly we're doing. So I'm a believer in the hype. So there's a couple of comments about the hype.
You believe the hype?
No, I think the hype, it's
a hype.
Yeah. So a couple of comments.
What's hype? The new drugs or the old drugs?
The new one. So the most likely individual to be cured of cancer seems to be a mouse, right?
Yes.
In a preclinical paper which led to a phase 1 trial.
Yeah.
Yes.
Right? Because every phase 1 drug cured all these mice.
Yes, it did.
Right?
But only 4% of those phase 1 drugs actually make it beyond phase 1.
That's the first thing I think that really does prove the hype. And we've had...
And we can find any mouse to find which drugs would be cured.
And so, TIGIT this, GITA this, LAG-12, there's a lot of hype.
Yeah.
And the mice all did really well, right? That's the first bit of hype. The second bit of hype, we've all got patients who have done really well and potentially been cured by drugs that didn't get further developed.
Yep.
Correct.
So I've got a patient I saw this week who was on a phase 1 drug, and I think he was the only responder on the study, right? The drug's been binned.
Alex and I have a couple of them.
He always says to me, why me? And I go...
You're lucky, mate.
Yeah. If I was smart, I'd know, but clearly I'm not smart. And to be honest, the drug company's not smart, because if they knew why he was, they'd go, this drug will help 3%, 1%, 10%.
Exactly.
We don't know. So that is the challenge of this hype thing. And I guess for me, I find the idea of looking at new agents really challenging, because the preclinical data, let's be honest, in every clinical trial that gets emailed to you, it's always the best one. And it's always curing the mice.
So I spend a lot of time actually looking at that and meeting with these companies. It takes a lot of effort and time, and I quiz and query, and you can pretty much get a sense of where this is heading and the science behind it, I find. I agree it is quite a bit of hype and the number of things that get past phase 1. However, I do think there is a reality that some things are looking fab.
So, Georgina, I was going to say, so you just going back quickly, you said are we using the tools we have at the moment to the best of their ability? And I think probably not.
Checkpoint inhibitors.
I think we're giving checkpoints unnecessarily when they
Yes.
don't work, obviously.
Yes.
We're probably treating people longer when they don't need it.
Yes.
Are there therapies like radiotherapy or other things that exist? Maybe chemo. I know outside of cancer, there's this, outside of melanoma, a big belief of synergy between chemo and immunotherapy.
I'm not a believer in that.
I'm not a
I
huge
think we're
believer
saving
either.
a lot of patients who need the chemo but probably don't actually benefit. But if the ones who benefit and are the tail of the curve on the immuno, they probably could have had doublet agent up first.
So
yeah.
for me,
but that's me.
that lesson of just because we had an old drug, any new drug gets added to the old one, I don't want seen repeated in melanoma. I don't want to see more trials of new drug plus Pembro in people who failed Pembro, or some drug plus PD-1 in people who failed PD-1.
Which
Why
we're
do
already doing.
Like,
We are doing the Carbogem plus or minus experiment
Exactly.
from lung cancer now.
It's just like everything has to
We
have
are there.
PD-1 in it.
We are
Why? PD-1 is not the problem in their immunity cycle.
Exactly.
So
I
I
agree.
think we've got to be smarter in terms of drug development.
So-
And hopefully some of these other newer drugs, which we're going to get to, are going to be separated from the checkpoints.
So let's get to that. So first principle, we think in cancer generally, in oncology, we could do better with checkpoints. We haven't maximised or optimised across cancer. That's the first principle. Matt's right, there's a lot of hype in drug development, a lot of hype. So the work is in weeding through what is hype and what's not, and understanding those mouse experiments and the mechanism and the science. But let's talk about some science. We don't have a lot of time on this episode. We're just going to go bang, 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 bang. Some of the highlights recently: cellular therapies, mRNA technology. And within mRNA technology, we've got some personalised vaccines, but it's not all personalised vaccines are using mRNA. And other immunotherapies you may want to highlight. So, for example, there are some great cytokines that are actually showing some activity. So let's start with cellular therapies, what they are and what we're seeing.
So I guess, checkpoints, just to recap, we know effectively are like removing the brake off the immune system. So removing inhibition to let the immune system do what it naturally wants to do.
That's kill the cancer.
Cellular therapies pioneered back in the 90s were thought to be the big breakthrough. At that point, highly selected patients, highly toxic, but could have durable control. Really the first real proof that immunotherapy could be curative. Basically, the idea is you cut out a tumour. If there are immune cells in it that we think are reacting against the tumour, you grow them up in a lab and give them back to the patient. So it's a bit like a transplant, an immune transplant, an auto-transplant of your immune system.
T-cell therapy, infiltrating T-cell therapy. And just one up on that, we're now using genetically modified T cells. So we take it out of the tumour, we grow them up, we genetically modify them, and then put them back into the patient. So that's all TILs.
So that's very labour intensive, very expensive. But if it cures people and cures them in a high proportion of patients
in one go,
in one treatment, can be appealing.
Yep.
But I think it's going to be difficult to see that rolled out for the masses around the world.
What about the other cellular therapies? So, for example, there's phase 1 data, fabulous cellular therapy where you plasma free. So you're not taking T cells from the tumour, you take cells, immune cells from the blood, then you genetically modify them with a PRAME so that they bind to PRAME, and then you feed those T cells back into the body, and that's showing some activity as well. So that's a cellular therapy.
TCR-T cells, right?
Yeah.
That's what
TCR-T cells. So what is T cell? TCR is T cell receptor T cells. Yep. Go ahead, make a comment.
I just want to make a comment on that. Even though I agree with what just Alex said, I think that's important to understand that one thing that Alex discussed was TILs, right? The beauty of TILs is that we take as Alex mentioned, the T cells that are there. So the T cells can really attack and identify a different range of antigens. So different types of molecules that are expressed by the tumor cells. The beauty of the TCR-T cells, as Georgina mentioned, so engineered, is that of course we can be very smart and active, or make the T cells very active and very good, but they are going to target one antigen.
Yeah.
So basically, that's a limitation. So basically, one thing that I want to discuss about this, I'm passionate about cellular therapy. I think that's going to be a big reality, and it's going to help a lot of patients. When I say a lot, even if it's 10%, if we know who they are, they become 100% for that. So biomarker is an important thing in this discussion. Having said that, we have to be mindful and understand exactly the nuances of each of the cell therapy. Are they going to target multiple antigens? Because one of the limitations in solid tumors is that our disease tends to be multiclonal, right? So it's not going to be polyclonal. It's not going to be one clone like hematological diseases. So CAR T cells tend not to work as well in melanoma or other tumor solids because of this. So that's
But there is the
one
theory of epitope spreading that we haven't touched on. So although you can make T cells with one T cell receptor aimed at PRAME, for example, that's the newest one at the minute, there may be a side impact on getting T cell recognition of other antigens in the tumour. But you make a good point that solid tumours like melanoma have lots of different antigens, and aiming at one usually does not
to me,
kill it
all of these early phase exciting things are proof of principle.
Yes.
That we have a way of treating cancers where manipulating T cells and often using T cells works. How do you get from that to the holy grail of an off-the-shelf product for everyone? Well, that seems a bit too simple. Or what Ines was alluding to, which is fine, multiple products, but more importantly for individual people. And I think that the concept of TIL is the signal that it's worth exploring. I don't know if in 10 years' time we're going to have cell therapy products that are very heterogeneous, but with all these extra products we can take the cured rate from 50 to 80 or 90.
Imagine you take your T cells, I think they're already doing this, you take your T cells, you plasmapheresis them, and you stick mRNA with neoantigens in it. Then you've got a whole stack of T cells against neoantigens.
In a perfect
That's
world,
happening.
But in perfect world, we may be doing that relatively quickly.
I reckon in the next 12 months.
Or it will be a hard grind, right? You go, right, we've got a treatment that can pick up another 20%. We don't know who those 20% are. So to find those 20%, we've got to treat all 100% with a treatment that's really toxic and costs $2 million each. I don't know which way we're going.
Yes, Matt, I'm worried, this is getting a bit political here now, but I'm worried that who is driving the bus here to a cancer cure in the immunotherapy world?
Georgina Long.
Is it clinicians seeing people in the clinic and seeing the need, or is it pharma trying to get a registration of a product and then go for it? Because as Ines has said, that's our
job, Alex.
No, I agree. But give me an analogy. Whether we have a different framework of vision in melanoma, but in other cancers, the beauty of targeted therapy is you find out who has the target and you can target it with a drug that works really well for them and does not work at all for everyone else that doesn't have it. We need an equivalent of that for all our therapies coming through.
But that's what Neo Irene is trying to get thinking. Neo Irene is doing the first step and saying, okay, everyone for PD-1 alone, off you go. Now we're going to focus on those. And then the next test after that will be the same thing. All of you who need triple or high-dose Ipi, you're done. Yeah. Now we're left with 20%. That's who'll get T cells. And then we need to pick out who's going to respond.
We need who they are early, rather than what I understand is happening at the moment is a first-line TILs versus PD-1 trial. Yeah. That is insanity.
And so I think the benefit the benefit of
we didn't open it, Alex, because we have that philosophy. You and I at MIA.
The benefit of your taking groups off is even if your treatment costs, let's say $2 million as an opponent. If that treatment costs $2 million, but only is required to treat X% and cures 90% of the X.
Exactly.
As a society, it's worth the $2 million.
That's what Ines was saying. She said we have biomarkers and then it's 100% if you know how to pick
when
them.
it's $2 million a pop and it has a 20% success rate, it's the number, whatever the financial equivalent of number needed to treat
Quality
is,
life,
it's $10 million a shot.
It's ridiculous because as a taxpayer, I would like my funding to go into schools and roads and infrastructure and not
curing cancer.
And curing cancer, not curing 1 in a million for $10 million when I could have cured some other disease for much less. You know what I mean? We've got to think as a community here.
Georgina, can you tell me about mRNA technology? What's that all about?
Oh, thank you, Chair. So I'm passionate about that. That is like a leap in medicine like we had with the World Wide Web and email, which unfortunately email is no longer something I love, but back in the 90s it was great. No, but mRNA is a massive technological leap in medicine. Basically, what we're doing with mRNA is we are taking certain code from our genetic code, we make code, inject it into someone's body, your body then produces the signal from that code, and that can be used, for example, for infections. So let's say measles, mRNA of measles, it codes a bit of measles, you express that measles, your body will make an immune reaction against that, you're vaccinated against measles. You can use mRNA for cancer. So for example, one trial we're doing right now, Alex, is PD-1 plus mRNA, which expresses PD-L1 when you inject the mRNA and IDO. And the idea is you will make immune cells against PD-L1, which is against tumour cells, and against IDO, which is the tumour microenvironment, to make it more immunogenic. And we've seen responses.
So
You've had a patient.
I would say I agree. So, but the examples you've used, are all, if you like, the really simple mRNA. The thing you're putting in your code is essentially to make a vaccine, be that COVID, be it individualized,
in
be
the
it
ones
PD-L1.
they gave you.
But
there are mRNA that are not.
No, and I think the coolest one, which was not melanoma, was reading something to show that you coded the whole drug in mRNA.
Correct.
And your body made the antibody.
So we did that. We've done a trial on that. We did a trial where the mRNA coded IL-23, OX40, and IL-36. And so your body makes those cytokines because you inject it. So you can vaccinate, make T cells against the protein you express with the mRNA, or your mRNA can actually make the drug. So it's like in vivo in vivo synthesis of drugs, meaning in the body.
And so
it's
I think
fascinating.
the challenge for mRNA, I think each part has a path. The personalised mRNA path. Mm-hmm.
So you mean personalised vaccine,
which is
now.
the one who's closest to prime time.
Yes.
The challenge is going to be cost, and how do we, a bit like TILs, how do you fund individualised treatment? But we can overcome that.
Can I just remind you the cost to do DNA? So what you need for that is DNA and RNA
which
sequencing.
Is cheap now.
Scale.
Yes, it used to be millions of dollars for one human. Now it's like $1,500.
And unfortunately, I think one of the fears of the barriers is, and back to Alex's politics, is in my experience, when you talk to a patient in Australia about an mRNA trial, they love it, right? Allegedly in America, it's poison. And so I think that will be the challenge for drug development, at least for this mRNA technology stuff.
Well, we'll develop them all in Australia then.
Done.
So personalised vaccines, just a quick word on that. You can use different platforms. mRNA is just one platform to make a personalised protein that your body can react against. You can also use peptides and you can also use DNA. So watch this space. We've got trials ongoing with personalised mRNA vaccines and also peptide vaccines.
And potentially we'll have registration trials reading out imminently.
They're therapeutic vaccines. They're not preventative vaccines. I think that's got to be said. So I think we've touched on some important areas in drug development. There are many more we could talk about. But any other last comments on that aspect?
I guess when we talk hype versus reality, it's amazing when, it wasn't that long ago that immunotherapy was 5 people sitting in a room at a conference sort of hoping, and now it's become the biggest thing in medicine. And, the fact that we can cure half of metastatic melanoma patients with one drug proves that immunotherapy can work. The fact it's curative, unlike chemo, target therapy, etc., means that is the goal. And I think there will get a point where we can cure the vast majority, maybe all patients, but the first leap has been the easiest leap. Everything from now is going to get exponentially harder, in my view.
Do you see a day where an oncologist won't use chemo?
I hope to see that. Whether it's in our lifetime, we'll see.
I hope so too. I
just
think
want
we're
to
going to
make
get
a-
there. Go,
a-
Inesh.
Yeah, very quick comment. So I just want to highlight on about this discussion two things. One is, again, biomarkers, really, that will be key. But the second thing is, in terms of drug development trials, we won't expand on that, but I think it's very important Alex made a comment about trial design is very important to select the right population, the right control arm. Again, if you put a PD-1 alone has control arm, we are already selecting the group of patients, as we discussed in other discussion of this podcast. But then the other thing is, for example, I'm just going through a new idea here. Why are we always combining with PD-1? Might make sense in some cases, but with vaccines, for example, what about ipilimumab?
Oh,
Low dose, since we are
CTLA-4
priming.
every
Try to understand
time.
the mechanism, right?
That's my
So in drug
thinking.
development,
Yeah.
so understand the mechanism, to understand so biology, but also biomarkers, they're all integrated. And it seems that sometimes we don't think enough when we design these trials. Just that final comment.
Agree.
And one other final comment just to go as well is there may get a point where we can vaccinate so we don't get cancer.
There will be. We haven't talked about that
That's another
today,
discussion.
but
That should
that's
be another
another
podcast.
discussion. That's another discussion. Okay, well, thank you all for that fantastic discussion. Thank you for joining us for Redefining Immunotherapy Lessons from Melanoma, brought to you by Melanoma Institute Australia and made possible by an unrestricted educational grant from Bristol Myers Squibb. We acknowledge the Camaragal people as the traditional custodians of the land where we have been recording today and pay our respects. This discussion was accurate at the time of recording, April 2026, but may not reflect the rapidly evolving treatment landscape and approvals in Australia. For more practice-changing education, sign up to our Melanoma Education portal at melanomaeducation.org.au. Thank you for listening.